This video chronicles the life of a soybean. This soybean is dormant right here. Dormant meaning it's dry, in a dry spot here. Can be cold or warm. The temperature isn't important, but this is not a dead seed. In other words, there is living material in here, but it hasn't come to life yet. What brings it to life? Well, when we push it into the ground, or when we just expose it to water, the seed starts to imbibe some of that moisture in there, and when that moisture gets in that seed, the wake-up hormone, which is a long fancy name but abbreviated as GA, starts increasing, and the ABA, another long name abbreviated here, starts decreasing. The ABA hormone was strong up in here during the dormant stage, but now everything is starting to change, and the proteins in this seed can actually start doing some stuff with just water. And the root starts to grow down here. But eventually, as that root grows and as the stem of the plant starts to grow, eventually you're going to use up all the food supply in this seed. So we have to come up with something else that is going to feed it. And the food that this thing drastically wants is the ammonium ion. And that also comes from plain ammonia. But we're not talking about man-made ammonia being pushed into the soil like you normally think. How does nature do this? Get that ammonia up here. Well, let's introduce a new character to the scene. Who might this be but a bacterium? A bacterium is singular, bacteria is plural. There's ammonia, the chemical formula for ammonia. And you see these little hairs sticking off of it and a tail, which makes it look like it can direct itself or move around. Is it plant? Nope. Is it animal? Nope. It's the special life form that bacteria occupies. What's this bacteria doing? There isn't anything for it to do until it finds a plant. So it works with a plant to produce the ammonia that the plant needs. What happens to the bacteria? Does it die in the soil? No, it's kind of like a seed in a way. It's kind of a dormant state. It's not too active doing anything. So let's see what happens if we can combine this bacteria or this bacterium to the root that just grew out of that seed and see what happens next. Here's the living bacteria in the soil. And here's the root of the soybean plant. This thing's a lot more mature. This seedling's pretty big compared to this. Before it got up to this stage, it's going to have some bacteria get involved. Now, normally you think of bacterias and infection and all that being a bad thing. But when we infect the root with this bacteria, it actually infects it, but in the process of infecting it, it creates this nodule, and then magically we can produce some ammonium ions that can be attracted into the cell wall of the plant. That's a little hard to see from this illustration, so let's go on to another illustration and see more of the chemistry of this whole thing. Let's walk through this. Soybean roots secrete flavonoids into the soil. What a strange name. Here's the name of the bacteria that detects them and releases nod factors. Another strange term. The root hair curls around the bacterium, or all these different bacteria, trapping them. It then allows an infection thread, a tunnel-like structure, to form, guiding the bacteria inward. Deep inside the root, cortical cells start dividing to form a new organ, the nodule. So this is like it looks like a cancerous growth almost on the plant, but this is a good thing. Once inside, the bacteria multiply and become bacterioids, a specialized form that can perform nitrogen fixation. That's what we want to talk about. This is the nitrogenase process we're trying to understand. The nodule contains millions of bacterioids. The plant supplies carbohydrates from photosynthesis. So that means the leaves are out there being exposed to the sun, and that gives us our energy source to do some of this process. The bacterioids use nitrogenase enzyme to convert atmospheric nitrogen into ammonia. But how does that actually get inside the cell? That's the next thing I want to show. These are a lot of details, but what I'm showing here is reminding us that the plant cells are separate from the bacteria. The bacteria are around those cells, but they're not inside the plant cells. And the plant cells are the ones that need the ammonium ion or the ammonia to form the amines and all the other amino acids. So how does that get in there chemically? Extracellular area up here is outside of the cell, and this is inside the cell. Well, what is this big structure here? This is the cell membrane, and somehow we've got to get the ammonia produced by the bacteria into the cell down here. So how does that happen? Ammonium is positively charged, and what happens is this membrane creates negatively charged areas. It's not ions, but it's a negatively charged area in the cell membrane that allows a tunnel-like structure, and this gets attracted by this into this cell, and eventually the ammonium gets down in here, and the cell can then make use of it as ammonia. Once the plant matures and dies off, then the root nodules are still producing ammonia, so that additional ammonia gets in the soil for the next crop season. Now, isn't that a nice thought? So soybeans pretty much fertilize themselves, and if you continue to grow soybeans in an area, it will pretty well fertilize itself. Now, it would be interesting to look at these bacteria and their magic with nitrogenase, and maybe that's where we can start next.